Bonjour à tous. On est très heureux d'accueillir aujourd'hui un ancien étudiant de l'IHU, Melhem Bilen, qui est maintenant à Beyrouth au Liban, qui est directeur de son laboratoire et qui est assistant professeur à l'université américaine de Beyrouth, qui va nous parler de ses travaux et ses perspectives sur le microbiote. Merci.
Merci Mathieu. All right. I'm going to be speaking in English because I'm really bad in French and just want to make sure that I'm delivering all the information correctly. So if there's anything that you don't get, just let me know. I can try to re-explain it or maybe try to find other words in French. First of all, it brings me a great pleasure to be back here, and I do recall my place that I used to sit here during all the WIP. And I'll be presenting today how I used the Culturomics approach that I was trained on here at the IHU in the United States at Stanford University, where I did my postdoc, in order to develop bacterio-therapeutic approaches to treat diseases that are gut microbiota related. So that's something that we all know, that the gut microbiota, the healthy one, is made of a diverse number of bacteria that live together at a certain relative abundance. And this synergistic way of living, and in terms of the number of the bacteria and so on, gives a healthy gut microbiota in the human body. Now, whenever there is a dysbiotic gut microbiota, which is like the absence of certain organisms or the presence of certain organisms, or a change in the relative abundance of certain organisms, was correlated to diseases. Now, the gut microbiota shaping starts from birth up until aging. It depends on the medication that we take. It depends on the food, the diet, and all this stuff. So it's highly affected by the way we live and the things that we take inside our body, because whatever we eat is whatever we are feeding the bacteria. Now, plenty of studies have been done on the gut microbiota role in health and diseases. And usually people rely on metagenomics, which is the sequencing approach to study all the DNA that can be extracted from fecal material, because we believe that the fecal material contain the majority of the bacteria that represent the gut microbiota. And they started comparing stuff between the healthy population and the diseased population, and they were able to find certain profiles of bacteria or relative abundance of organisms, absence or presence of certain organisms. And the examples of diseases that was correlated to the gut microbiota are the multiple sclerosis, non-alcoholic fatty liver disease, obesity, type 2 diabetes, of course the Clostridium difficile infection, and so on. So plenty of disease and a lot of correlation with the gut microbiota. Now, the beauty of the sequencing approach is that it allows sometimes to detect the presence or absence of certain organisms that we can use in mice models in order to prove the correlation of the gut microbiota and the disease. Like, for example, the Faecalibacterium prosnitzi in IBD, Akkermansia and obesity and all these things. So whenever we have certain diseases, metagenomics would allow us to understand these, the presence and absence of these organisms. However, metagenomics has plenty of drawbacks, and these drawbacks can be because of the depth bias. 1 gram of stool can have like 10 power 14 cells, and there is the low abundant organisms that can have key biological roles, but it remains undetected, and so on. And it's beautiful to have results on an Excel sheet, but we actually need the living organisms in order to develop bacteriotherapies and to really use our information and to help treating diseases. Now, the success of the fecal microbiota transplantation back in the time to treat the Clostridium difficile infection motivated most of the scientists to try to dig in further the gut microbiota, specifically finding a way to manipulate this and to cure diseases. The problem with the fecal microbiota transplantation is sometimes the inability to control the content of the fecal material itself. So there could be certain pathogenic organisms that can skip the sequencing analysis, and there has been like plenty of death cases in the FMT, especially the multi-drug resistant E. coli that infected patients and ended up with a death case. At the same time, certain toxins that go undetected in the fecal material. So there is, it's an uncontrolled environment. It has the potential to treat, but it is uncontrolled. And if we cannot control it, it means that we might have risk of, instead of helping the patients, killing the patients. So the common strategies now to, in most of the labs, to study the gut microbiota and how we can intervene in order to treat diseases is by performing fecal microbiota transplantation to the mice. So we get germ-free mice, and then these germ-free mice, we gavaged it by stool samples from human donors that we assume that they are healthy, and we consider this mice as a humanized mice. To study disease models, we take germ-free mice with certain genetic mutations, or we can induce certain profiles inside these germ-free mice, and we can also put, we gavage these mice with fecal material and try to study the disease development or the treatment of this disease by doing a backfill experiment and so on. So usually this is the approach. Now, again, we remain in the same problem of humanizing mice experiments, that we are still giving and studying samples that are undefined and we cannot control. So there has been interest in creating bacterial communities that we call synthetic bacterial communities. And there is plenty of startups now in the United States and even in Europe trying to create bacterial cocktails for specific treatments. Example is the Vedanta Bio that created, I think it's Vedanta, that created the complex to treat Clostridium difficile infection, so the use of certain bacteria to give it for the patients and treat them eventually. So most of the synthetic complexes that have been out in the market, not in the market, but basically in the research and being studied for clinical trials and so on, they are made of 1 to 3 organisms, up to 14 organisms, 15 organisms. And this is not also representative when it comes to the gut microbiota, because if we want to enumerate the number of bacteria by culture, we can obtain around 200 bacterial organisms from the same fecal material. Metagenomics, we get 347 on average. We can get 237 if you are doing the 16S instead of the shotgun sequencing. So making a bacterial complex with small number of bacteria to treat disease is also considered non-representative, because we might be causing a dysbiosis and we are not refixing the whole microbiota inside the gut. Now, this work led to the creation of the Microbiome Therapy Initiative at Stanford University, where Professor Michael Fishback, along with Alice Chang, before I went there to my postdoc, they decided to create the most complete synthetic bacterial community that resemble by its content and its function the fecal material and the human gut, the healthy human gut, and use this to modulate it to treat diseases. Now, by creating a synthetic community, we will be able to perform dropout experiments where we move certain strains to make sure that this strain play a role or doesn't play a role, has a therapeutic potential or not. So synthetic microbial community was the big thing, and it was the hypothesis of what we wanted to do in the US. Now, in order to create the synthetic community, we need to make a list of these bacteria. Now, what Alice did was she went to the Human Microbiome Project, where they have deeply sequenced fecal material, and she chose the most prevalent organisms among all these stool samples. And not the relative abundance, the high in relative abundance, but the most prevalent. So we assume that this is part of the healthy gut microbiota. It's prevalent everywhere. So let's just take this list and test it. So it ended up having 99 strains, and all these 99 strains were purchased from strain collection banks. And I recall also when we wanted to add Archie, we purchased the Methanobrevibacter from here, from the YASU, from the CSUR. So the most prevalent bacteria and the healthy gut and the healthy fecal material collected in the HMP project were the candidates for our synthetic community. And she added a couple of organisms, which are the Ruminococcus bromii, because it has the potential of polysaccharide digestion, and the Clostridium sporogenes, because it has the potential of gene editing in case you want to perform engineered microbes for specific functions inside the synthetic community. 3 alternatives from this list that we obtained from the HMP were added because we failed to obtain it from strain collection bank. And we called this first list as HCom1, okay, Healthy Community 1, that is made up of 104 strains, assuming that it represents the healthy gut microbiota. Now, to test the, so before I want to emphasize on that we purchased these organisms from the strain collection bank, and I was discussing with Mathieu that if we want to create a bacteriotherapy that is FDA approved, we need to isolate these organisms from a fecal material collected from a healthy human and be able to control it back in the history. So even if there is like all the organisms in the strain collection banks internationally, we need to isolate these organisms. And this is the importance of culturomics when proceeding, because we really do need to apply this in order to get there. Now, to test the stability of the community that we have, because it's not easy to obtain a bunch of hundreds of organisms and live together, because most of the organisms, they will compete. Some of them, they will be excluded naturally or included. So it's going to be a mess, culturing 104 organisms together. So there was an experiment that has been done in vitro to prove that these organisms can survive together. And within a span of 48 hours, these organisms can survive at a certain relative abundance that remain stable all the time. So we start by a random concentration, and then whenever they are put together, they reach a stable relative abundance. Of course, we needed to do technical and biological replicates. These technical and biological replicates, they serve the confirmation of our strategy and capacity to replicate this in the future in mice models and so on, and the stability of this bacterial complex that we have. So after 48 hours, we have a decent relative abundance distribution of all the organism, stable one. We have biological and technical replicates that resemble the same. Now, the strategy to test these communities, if they are capable to colonize the gut system, we used germ-free mice models, and we cultured these organisms independently in a general medium that was modified in order to contain all the nutrients needed to grow these organisms. So these 104 organisms, they were cultured in an unnormalized concentration, so each has its own OD. And after that, we normalized the concentration of all these 104 organisms to have the same OD. Then we mix all these together, and after mixing it together, we take the germ-free mice and we do a gavage. So what we have seen is that over 4 weeks, the relative abundance of the organisms change, but at 4 weeks, it becomes stable. So if we keep on sampling, taking fecal material from the mice, after 4 weeks, we do see that the relative abundance of the organisms, it remains the same. So these organisms, they just colonize naturally and organically inside the gut of the germ-free mice. Now, for the analysis that has been done, there was a software called NinjaMap that was developed at Stanford University by the bioinformatics team that is capable of detecting organisms at 6 orders of magnitude. So even low abundant organisms can be detected. And also MIDAS, which is also a publicly available tool, it was used to detect other organisms in future experiments that I'm going to talk about in a bit. Now, to see if these organisms colonized or not, we performed metagenomic sequencing at week 4, and 101 organisms out of the 104 chosen, they colonized, except 3, which are the ethanolygen, the Clostridium methylpentosum, and the Ruminococcus albus. Now, the theory is that, are these colonizers or not colonizers? I think it's a question of the ability of these organisms to be grown in vitro in the lab easily. These are hard organisms, fastigious organisms. So sometimes we might have skipped a step in growing them to a level of colonizing level. So so far it haven't colonized. Maybe it's the problem of not being able to obtain a high level of viable colonies, of viable cells, or they are just non-colonizer among the other community. Other than that, all the 101 organisms colonized. And again, I repeat that it's not easy to have 100 organisms living together and being able to make them colonize as well without any out competition. Now, the results of colonization, it was among different cages, among all the mice that were tested, it was amazing. They all showed a similar relative abundance distribution of the organisms, making it like really a magic. We take these 100 organisms, we put them in any germ-free mice, and they will end up colonizing at the same relative abundance after 4 weeks. So this is the first step where they said that, okay, we are capable of creating a synthetic bacterial community, and this synthetic community can colonize germ-free mice. And it's a controlled environment, of course, because we know each strain and the profile of each strain. As for the stability, as I mentioned, that the community reaches in germ-free mice a stable configuration after 2 weeks, and after 4 weeks, we see that everything resembles in different cages, and it remains stable for even different generations. Now, we agree that why choosing 104 organisms out of all the organisms that are present in the HMP project and all these studies. So we needed to create a beta version of this community. And the beta version of this community, to do that, we didn't go and looked in the HMP and tried to add organisms. We said that if we do a fecal microbiota transplantation to a germ-free mice that was already gavaged by a synthetic bacterial community, the HCom1, we assume that if there are organisms capable of colonizing that are missing, they will colonize. They will fill ecologically the niche that they belong to. So this is the theory. We took the 104 stocks that we purchased from strain collection banks. We cultured them at an unnormalized condition. We normalized it to receive a common OD, and then we gavaged germ-free mice with it. And then after 4 weeks, because we know that this community after 4 weeks, it reaches a stable, it reaches a stability with a stable relative abundance, we performed a challenge, a backfill experiment. It's a fecal microbiota transplantation by using a human fecal material. So we took healthy human fecal material and we gave it to the mice. And the hypothesis was that there are going to be excluded invaders, organisms that are present inside the fecal material that will not be colonizing the gut of the mice. And there will be colonizing invaders. These are bacteria that has to be part of these HCom1 and should colonize. Now, after 4 weeks, after an additional 4 weeks, we sacrifice the mice, we collect the fecal material, and we see what are the organisms that jumped inside the community and what are the organisms that were excluded from the community. So, when it comes to the strains that remained intact from HCom1, they were not excluded. They kept a proper relative abundance comparable to that before the gavage. So before the gavage and before the backfill and after the backfill experiments, the strains that are part from HCom1, they remained with the same relative abundance, the ones that stayed and were not excluded. And there were a bunch of organisms that jumped in. And we assume that these invaders were the missing organisms from the HCom1. Now, 89% of HCom1 stayed in the gut. There was 11% that jumped in from the fecal material that we used for the backfill experiment. And now we wanted to collect these organisms and try to make HCom2, which is the better version, and see that HCom2 if it's a complete now community or not. So we wanted to add the strains, and mainly 24 strains, and that's not a lot. compared to the number of bacteria that can be found in the fecal material, meaning that we have reached almost a perfect community, but we had 24 that were missing. Now, we were able to purchase 22 organisms. We had 7 species that failed from HCom1, that failed to colonize, so they were excluded. Now, 97 strains that remained from the 104 organisms originally chosen for the HCom1, along with the 22 new strains that we purchased from culture collections, and these are the 22 that colonized after the Backfill experiment, allowed us to create an HCom2, which is a new version of HCom1 with a total of 119 organisms. So, when we took HCom2, we did the same experiment. We grew it in vitro. We normalized the OD, and now we put it in the germ-free mice. After 4 weeks, we also tried to see if this community colonized or not. When putting the HCom2 inside the germ-free mice, they reached a stable configuration of 100 of the 119 strains colonizing. So 100 of these 119 colonized, and they reached a stable configuration in 4 weeks. So this version of 100 organism is now the new, most stable version of synthetic bacterial community. And all the HCom1 strains that colonized also had the same relative abundance scheme as in the first experiment where we just used HCom1 for germ-free mice. Now, we did also a technical and biological reproducibility test to confirm that these organisms, they can always colonize the mice from different way of preparation. I think you know the difference between the technical and the biological reproducibility, and HCom2 had a great technical and biological reproducibility in germ-free mice. Now, to make sure that HCom2 is like the human gut microbiome, it is robust to changes in the host genotype, we decided to change the type of mice that we used. So we were using the Swiss Webster mice, germ-free mice, and we chose to another category of germ-free mice, and they ended up having the same profile of HCom2, which means that it's like the human body. Whenever we put the fecal material in it by the FMT, it is capable of colonizing regardless of the individual. So now at this point, there has been also other backfill experiments to test if there are stuff that will jump into HCom2 or not, but we saw that the HCom2 is pretty stable. It doesn't allow invaders. It colonizes these 100 organisms, colonize very properly inside the mice. And now we wanted to test if these strains, they resemble, if when colonizing the gut of the mice, they resemble functionally to the gut microbiota of the mice that have been challenged by fecal material from human sources. So we took a group of mice, we put HCom2 in it, and we take a group of mice, we put fecal material in it. And now we want to compare the functions, metabolomically, immunological, relative abundance, presence at the phyla level of the most abundant organisms, and so on. Now, to do that, germ-free mice, HCom2, another group, germ-free mice, but fecal material. We harvest the immune cells at week 2, and at week 4 we collect urine and feces for metabolomics and feces for plating for colony-forming units to make sure that we have the same approximate number. Now, after 4 weeks, we collected the 2 groups. So HU1, HU2 are the group of mice that were gavaged by human fecal material, healthy human fecal material. This is another donor. We have HCom1, which is the original community that we did, HCom2, which is the new community. And of course, there is the germ-free mice and the other ones. Now, whenever we wanted to count the colony-forming units by plating the fecal material, we see that the number of colony-forming units from the feces collected from mice at week 4 resembles the one of the humans. So humans and HCom1 and HCom2 had the same number of CFU. As for the relative abundance, we saw that the ability first to do technical and biological replicates, it was also the same, and for both the fecal material and the synthetic communities among all the mice. So CFU is the same. Now we come to the immunology profile. To do so, we collected we stained and extracted the colonic immune cells, and we did an assessment by flow cytometry. We wanted to measure the CD4 plus T cells, the IgA plus B cells, the macrophages, CD11 B plus dendritic cells, and the monocytes. Now, whenever we take into consideration the humanized mice and HCom2 mice, they have the same thing. So they have the same immunological profile. When we give the mice the fecal material from human sources, and when we give it with HCom2, we collect these cells, we extract it, we assess the number of these, they have a similar immunological profile. Now, when it comes to the metabolism profile, fecal pellets and urine samples, they were analyzed using targeted metabolomics, and we checked for aromatic amino acids, metabolite levels in the urine, and for primary and secondary bile acid level in feces, and HCom2 colonized mice and the humanized mice, they had the same profiles whenever we plotted. And at the same time, we can say that HCom2 can perform at the immunological level and at the metabolic level similar to a humanized mice. Okay? So now, instead of using fecal material that we considered undefined community, uncontrollable, to humanize mice and use it as mice models for future bacteriotherapy development, we have developed the concept of creating a synthetic microbial community that is capable of colonizing germ-free mice in a stable way. We can obtain biological and technical replicates, and it can act at the immunology and metabolic profile the same way as the humanized mice. So far this is perfect. The synthetic microbial community concept is established. Now we want to see if the synthetic microbial community can act similarly whenever there is pathogen invasion to the germ-free mice that were colonized with human sample, the humanized mice. So Alice chose the enterohemorrhagic E. coli, that because it's a life-threatening diarrheal infection, it causes hemolytic uremic syndrome, and enteric colonization by other E. coli strains, and that is correlated to malnutrition and IBD. In the HCom2 community, there were no Enterobacteriaceae, and there was only 3 species of proteobacteria, which means that there is no resistance by close relative occupying the same niche whenever we introduce the EHAC, the enterohemorrhagic E. coli. So to check the ability of HCom2 to create a resilient gut microbiota inside the mice, similar to the human fecal material, we performed an experiment where we used HCom2 in germ-free mice. And some of them, we added the Culturomics, which are a bunch of organisms of E. coli and Enterobacter that increases usually the resistance toward the EHAC, the enterohemorrhagic E. coli invasion. And we used other communities that were published, 12 bacterial organisms that were published and known to be resilient to the EHAC, and we assessed it along with comparing it to the human mice. So to do so, synthetic communities or the community HCom2 or the community that was already published made of 12 organisms or the human fecal material, they were used to colonize germ-free mice. And then after 4 weeks, because it reaches its relative abundance stability, we challenged it with EHAC, with the enterohemorrhagic E. coli. And then after 4 weeks, we started sampling and checking the level of the enterohemorrhagic E. coli to see if this HCom2 can provide protection against this pathogen. So there has been EHAC clearance in the HCom2. Whenever we look at in blue the HCom2, we see that the number of EHAC is similar to that of the stool of the mice that have been gavaged with the human fecal material. Now, if we want to add the Culturomics that is known to have bacteria that also support the resilience of the community inside the gut towards the invasion of the EHAC, it acted approximately less efficiently than HCom2, or we can say similarly. So this is the community that has been published already to be able to clear the EHAC. We see that HCom2 is capable of performing the same way. Whenever we add the EM, it's also even better, and we can see that the EHAC goes down and it's cleared. All right, so we can take as a conclusion that HCom2 can act similar to the fecal material when it comes to robust resilience against EHAC. And at the same time, the enteromics does help, but also the HCom2 by itself is good. So it's sufficient, complete, and exhibit comparable level of colonization resistance to a native fecal community. So this is what we wanted to check at the end. If this fecal material, if this community can form a gut microbiota environment similar to the human healthy gut microbiota that is resilient to colonizers and to pathogens that can invade. To confirm these results and to show that it's the synthetic community itself that is protecting against the EHAC colonization, we wanted to make sure that the architecture of this community is complete by performing dropout experiments. We decided to remove firmicutes, then the varicomicrobia, then the actinobacteria, then the proteobacteria, and we reperformed the experiment. Just to say that the HCom2 is complete by the presence of all the organisms and not by part of it. It's the completion of this set of strains that colonize is what giving the protection and the resilience of this community and the gut of the mice against the pathogens. So when we drop the firmicutes, we can see that EHAC is not cleared. Also for the varicomicrobia, actinobacteria had no real effect, but it also had, compared to the HCom2, it wasn't that good. Proteobacteria. So anything that is dropped from HCom2 will make the community non-resilient and sensitive to invasion, which means that HCom2 by itself is a almost complete synthetic community that performs resistance towards pathogen invasion. It has a similar immunological profile as the human fecal material, same as for the metabolism. So this is great. Now, the synthetic microbial community, the complex one that is made of hundreds of organisms, the model has been established. It's a defined model. It's a controlled model because we know the genomic profile of each organism that is used. We know how to clear it if in case we face any undesirable effect if using a synthetic community. So it's a controlled environment, not like the fecal material. It is stable and it is reproducible. Every time we put it in germ-free mice, it ended up, all the bacteria end up living at a certain relative abundance, and it takes a stable configuration. So given this model, now we are capable of creating a similar thing that can be used to treat diseases. So instead of the FMT. So we already said that it resembles the human microbiome at the immunological profile, at the metabolism profile, and also by its colonization capacity, resistance to pathogens, and the relative abundance of organisms compared to the fecal material is the same. Now, in order to start working on disease models and to create bacterio-therapeutics, we need human isolates because this is the regulation of the FDA. I cannot just go and order, let's say, Faecalibacterium prosnitzi that was isolated from, from animal sources, if it exists, just a random example, or any other bacteria, and say that, hey, I'm going to use it and try to manipulate it and give it to the patient. This is forbidden. So we need the organisms, and that's where we need the culturomics. And because it allows us to isolate fastigious organisms, and I was surprised by the lack of professionals that are capable of culturing fastigious organisms and anaerobic organisms, and how important it is, and how much people in different labs in the United States, they were ready to give it all to be able to just create a strain collection bank that is FDA guideline compatible, because it's going to open endless doors to develop bacteriotherapies, startups, and also benefit the whole community overall at the scientific and medical level. I'm not going to be talking in details about Culturomics because I guess everybody knows about it. Culturomics was established here at the HU, mainly by Lagier, Mathieu, and all the older generation, under the supervision of Professor Raoult. And the main theory or hypothesis was that any organism can be cultured as long as we find the right route to culture it. So there is nothing called uncultivable organisms, because back in the time they said that 80% are uncultivated, and 80% of the gut microbiota are uncultured and cannot be cultivated. Culturomics came to prove that this is wrong. We can do that. And the importance of this, of being able to culture these, is now very clear, because we need this technique, we need this approach that can, so far at this moment, cannot be replaced by any artificial intelligence or robotic engineered instruments to isolate these organisms. We still need to do this in the same old school fashion, and it's needed by most of the people. And metagenomics, as I mentioned, is a great approach, but it will only give us reads and sequences that we can see on the screen, but we can never create a bacteriotherapy with it. The drawbacks, we already talked about it, the presence of an incomplete genomic database, and Culturomics play a big role in this, because whenever we try to optimize HCom2 by checking other organisms, we see that there's plenty of novel species isolated here from this lab here that are part of the commensal flora of the human gut microbiota, and is needed in our community, in our bacterial community, to establish a healthy gut microbiota. Alistypus senegalensis is one example. All right. So the incomplete genomic database that is being overcome by Culturomics, and mainly here by this lab that has more than 500 novel species, is allowing us to keep on optimizing the bacterial communities and knowing what are the organisms that are part of our normal flora. Sometimes the inability of detecting pathogenic species and not having live biological organisms to proceed. All these are just like drawbacks of the metagenomics. But it's important to say that only Culturomics is able to overcome the main drawback of metagenomics by providing whole genome sequences and 16S sequences and reference stuff for the novel organisms that are isolated, which are part of the gut microbiota and needed to treat diseases. And the importance of the Culturomics was very clear by this beautiful paper that was published here as well, where only 690 organisms were known before the introduction of Culturomics to be isolated from the human gut, and it escalated to 1,761 in 2018, with around 380 novel organisms. Now, Mathieu told me that you guys reached the 500 organisms, which are all key for the development of bacteriotherapy. I left Stanford University with 3 ongoing projects. I cannot really report the data because there is ongoing clinical trials and stuff, so I respect the non-disclosure agreement and the confidentiality of the data over there. But there is now a microbiome therapy initiative that is led by Professor Michael Fischbach at Stanford University in the ChemH department that is part of the Faculty of Medicine, and the main objective is to create defined microbial communities to treat diseases similar to what I presented for HCom1 and HCom2. We have projects that are ongoing with the Gates Foundation to also create microbial communities to treat the Pakistani population that are suffering from malnutrition. And I also worked a bit on the PD-1, PD-L1 blockade therapy, because it's known that there is also a paper that has been published with the Wargo lab, Jennifer Wargo, with whom I was collaborating as well, on the role of the gut microbiota and the response towards the immunotherapy treatment. And there was a collaborative work with Yashu here. No, Mathieu? I can't recall with the Wargo lab. There was... Okay. But Jennifer Wargo had, I think it was one of the authors, no?
No, not in that work, but
okay.
It was a back-to-back paper.
Okay. So overall, I just wanted to highlight that Culturomics, although it seemed simple and repetitive work, it ended up being an essential work to develop to develop real treatments for diseases that can be used for clinical trials and clinical assays. And for now, I think that there is plenty of potentials to treat diseases via the gut microbiome, and the concept of synthetic community will bring a lot of prosperous treatments in the future. That's it. Thank you.